everyone, and welcome to the third day of the Illumina Genomics Forum. The past 2 days have been amazing and definitely a call to action to enable genomics for everyone, every person on the planet. To do this, we must collaborate better, be better stewards of these technologies by sharing information more broadly and stepping into leadership roles to unlock their potential. We've waited decades to get to this point. We are in the genomics era. And this cannot be available just to the rich and lucky, like we heard earlier. Bill Gates has devoted his work to changing people's lives, to bring equity and health across the world. As co-chair of the Bill and Melinda Gates Foundation, he shapes strategies and advocates for issues that further the foundation's goal of improving equity in the United States and around the globe. Through his Gates Ventures office, he pursues innovation in technology and healthcare, including Alzheimer's research. Bill also founded Breakthrough Energy to support the next generation of entrepreneurs, big thinkers, and clean technologies to address climate change. This morning, we have the pleasure of hearing Bill talking to us about what genomics helps us see, turning potential into progress for a fairer world. Afterwards, I'll have the pleasure of joining him for a short discussion. And without further ado, please join me in welcoming Bill Gates.
Well, good morning. Obviously, I'm not a genomic scientist or a biologist, but I am thrilled to be here and talk to you about how genomics is enabling the Gates Foundation to achieve its very ambitious goals. Those goals are to make it so that a child born everywhere has the same chance of surviving and thriving. And so what I want to talk to you about is how we're using genomics to take a disease like malaria and track it and eventually be able to achieve malaria eradication. We're using genomics to solve malnutrition, understanding for the first time what's going on in the guts of those children and why they grow up with neither the physical or mental development they deserve. So the foundation got going about 22 years ago, and I was really stunned at that time to learn about how little was going in to helping with these diseases and how little we knew. We didn't know, when you died of diarrhea, what the pathogen was. We didn't know if you died of pneumonia, what the pathogen was. Particularly in the first 30 days, we didn't understand what interventions might have been able to save a child's life. And so in every one of these areas, it's the genomics revolution that's created the platform that allows us to gain those understandings and actually come up with new interventions and being able to use the right intervention at the right time. Also, I want to talk a little bit about the pandemic in genomics. The pandemic was an incredible tragedy, but I think PCR diagnostics was one of the few bright spots along with the vaccines. And if we use that in a more intelligent way, we should be able to stop outbreaks before they become pandemics. So a lot of innovation and having that innovation get less and less expensive allows it to be even more aggressive in using these tools. So what's the report card on global health? Well, since the turn of the century, we made unbelievable progress. The number of children who die under the age of 5 was a little over 10 million at the turn of the century. And now it's under 5 million. We had a goal to cut it in half again by 2030. In hunger, because we're improving crops and seeds, that was cut by a third. Polio, which is attempting to be the second significant disease eradicated after smallpox was eradicated all the way back in 1980, has reduced the caseload by 99%. Now, sadly, for all this progress, progress in malaria deaths, HIV deaths, TB deaths, all cut in half, despite that progress, ever since the pandemic, we've actually either stalled or gone into reverse. In fact, if you take the broad set of goals that the United Nations calls the Sustainable Development Goals, and those were goals that were set in 2015 with the aim of achieving them by 2030, we are not in good shape. Last week we had the General Assembly where we get together and actually look at that progress, 7 years into the 15-year period, and what we see is we're behind on all of those goals. Even the ones we were on track for before the pandemic, we no longer are. And so we have to accelerate innovation. The only way that we're going to get back on track is a combination of better tools, where genomics plays a big role, better targeting, where the surveillance enabled by genomics plays a big role, and we can achieve those goals. In some cases we'll be 4 or 5 years late, but the idea of cutting childhood deaths all the way down to 2.5 million, which gets us very close to an equitable outcome, we need to rededicate ourselves to that task. Now, ideally, we'd always be seeing the viruses and bacteria that are circulating. That kind of understanding, including for respiratory disease, which is your major risk for a fast-spreading epidemic, that's something that we don't have today. Overall, the rich world, in a sense, you could say it lost its focus on infectious disease, because if you just confine yourself to the rich world, it's not a major source of either morbidity or mortality. If you turn to the developing world, the other countries, infectious disease is still responsible for more than 4 in 5 deaths. And so whether you care about surveillance to avoid pandemics, or dealing with the pandemic, or dealing with those infectious disease burdens that are still quite large, although very much out of sight for the rich countries, you need genomics as a very big tool there. Let's talk a little bit about malaria. Malaria is still killing 627,000 people. That's down, as I said, by a factor of 2, because we do have bed nets and better drugs. But malaria is a tough one. It's constantly evolving. The mosquito that carries malaria is evolving. There's many different species, and we have, of course, vivax, falciparum, different forms of malaria. And we always need to understand where we're getting drug resistance, insecticide resistance. We can only target our malaria control programs if we see all of that data. And so now there's been really great work in sequencing some of the mosquito genetics and the plasmodium, the pathogen genetics, so that we're constantly understanding how prevalent is it? Was it imported from a different area? Is there a new threat going on? In fact, what we're seeing is that there is now an Anopheles mosquito, Stefansi, that for the first time is a vector that survives quite well in urban areas. And so that's been very helpful to us to see that spread and now get to that targeting. We are going to use genetics, a gene editing construct called Gene Drive. We have lab level trials for that. It's a way of making sure a trait is inherited from either the mother or the father, so you can drive it into the population pretty dramatically. So things like shortening mosquito female lifespan, which will be a very key tactic as part of our malaria eradication. All of our diseases can benefit from this constant database of genome tracking. So for example, with TB, we need to understand how it's spread. We need to see what drug resistance is emerging. In that case, we do have a great pipeline of drugs that use different mechanisms, so we ought to be able to stay ahead of that. Likewise with HIV, understanding transmission patterns and being able to know when you need to change that first-line therapy. We also saw this in our polio work. We sequence all the samples we get there, and that has helped us see even the tragic fact that the polio virus, where we only have 2 countries that we've never eradicated now, which are Pakistan and Afghanistan, now it's spread back. We're seeing it in London, we're seeing it in New York. And so it reminds us that if we have polio anywhere, it could get back, and so we really need to get to 0 as quickly as we can. Of course, another great leap we're taking is taking the genomic data and being able to map that to proteins and protein shapes. And of course, all of you know the incredible potential that provides. We've got Rosetta fold, we've got alpha fold, and as we put more data into these systems, particularly data that is extremely precise in terms of the shapes, down ideally below 1 angstrom of uncertainty in terms of the positioning, that gives us tools to do drug design. We have examples of that even with one of the COVID vaccines, understanding nanoparticle assembly through in that case, the Rosetta Fold tool made a huge difference. It's a vaccine that was just invented at the University of Washington Institute for Protein Design, and now it's gone all the way through the process. It's one of the licensed vaccines, and we're looking at, it may have unique characteristics in terms of the breadth of protection and duration. And of course, although the vaccines we got were a miracle, they still fall short in terms, they don't protect you for decades, they don't block infection, and they're not universal in the sense of taking the entire coronavirus family and creating antibodies that are broadly protecting. So we have a long ways to go to get the ideal vaccine, and genomics is how we get the information, understand, okay, what are these antibody populations, what does this breakthrough look like, and feeding that into these shape-space tools that will only get better. After the pandemic, I wrote a book about trying to do a better job next time. Given the fatality rate of this pandemic, we actually did a pretty poor job in terms of blocking it. Some countries have dramatically lower death rates than others, and so there are some best practices, but our general surveillance capability is very, very limited. And so in that book, I talk about how we have to be out there sequencing on a regular basis lots and lots of things. The idea of sequencing sewage, that turned out to be very valuable. We've been using that for decades for polio, works for cholera, typhoid. Amazingly, you wouldn't think it would necessarily work for a respiratory virus, but in the case of COVID, it actually gives you the best, almost real-time signal, not just of prevalence in your community, but also a sense of what the various variants are. The foundation was running a thing called the Seattle Flu Study, where we were taking samples of everybody who had respiratory systems and just trying to understand over the course of the year what are the populations of the various respiratory viruses, and getting a sense of how those are spread, which is very important as you want to have quarantine techniques that do the least damage, understanding the age structure of that transmission, the location of that transmission. And in the future, I think that kind of respiratory surveillance, we need that on an ongoing basis. It shouldn't just be a one-time research project, which in that case happened to be the first place that we detected that COVID had spread well beyond the people who'd actually been outside the country. So the idea of what they call reflex testing, taking some of those samples and going all the way to see is there a new variant or even a new respiratory pathogen that we haven't seen before, that should be done on an ongoing basis, and we can only do that because of genomics. So, surveillance, we're very underinvested. Having that kind of data would have let China or wherever the initial outbreak is see it and warn people. The actions you take at the very beginning are what make a huge difference, because otherwise you get exponential growth and geographic spread. And there are scenarios that are kind of negative to focus on, but something like bioterrorism, where even beyond the type of surveillance you would want to do to detect a natural pathogen emerging, there you'd really want to take the air, the environmental samples, a variety of sources, particularly at places like airports, and really trying to understand what's going on. So it's fantastic that those tools are getting to the point where that will eventually be common sense. In parallel, of course, to come up with countermeasures, all these proteomics understandings, where there's a lot of open source information being put out, that should allow us to have libraries that are either close to the pathogen or can easily be modified to have high affinity to the pathogen. And so our ability to intervene more rapidly with antivirals, antibodies, that's something to be very optimistic about. The gene sequence is the basic starting point that we have to have, both for the non-pharmaceutical interventions where we have quarantine policies, but also it's kind of the starting line of building the interventions. Eventually the vaccine is in there, but there's others like respiratory mini proteins to block transmission that can probably be developed even faster. Well, let me move on from pandemics to talk about nutrition. We've never really understood nutrition. Some kids in developing countries, even twins in a family where you're getting exactly the same diet, one of the twins will have stunting and never fully develop, both mentally and physically. So looking into the microbiome and understanding what's going on there, and very complex interactions between what you eat, the drugs you take, the immune system, and this microbiome, we are finally gaining those understandings. Obviously, we couldn't do this without cheap sequencing. There's trillions of microorganisms living inside us, and so even to get a statistical sense of, okay, which ones of these are associated with good incomes like B. infantis and which ones are associated with inflammation and bad outcomes, we're only beginning to gain that understanding. The Economist had a great piece this week about how the foundation is doing nutritional interventions where we make sure that we're giving the kids foods that encourage the good part of the microbiome and discourage the bad part. And we're seeing that that actually is associated with higher growth. And so it's very, very promising to take malnutrition, which in Africa, over 30% of kids, as they develop into adults, malnutrition means that they are way behind. And even if you do have an opportunity to educate them, they're not going to achieve their potential at all. So by looking at the microbiome of infants, we really have a sense of, okay, what is good gut health? I read recently people are going to look at microbiome around cancers as well. Well, that's a very promising thing. We also see that affecting the microbiome in the sexual organs, that we may be able to reduce HIV transmission pretty dramatically. And so these bacteria and understanding them, it's something we've been blind to, but now that we're not, that is very, very promising, because in the case of nutrition, we're seeing that these interventions could be very, very cheap. Ideally over time, we really understand the human infectome. We understand the pathogens that are out there, how they spread. If they evolve in a way that's not good, we understand how you block them. When COVID hit, our disease surveillance was very, very poor. Likewise with the microbiome, as we're building up an unbelievable amount of data about that and connecting it with what's a healthy diet, that will lead to help, not just for malnutrition, although that's the foundation's priority, but also some profound insights that will help with overnutrition, which plagues the rich world and many middle-income countries. Now, we do need to make sure, as we're going about all this genomic work, we're doing it in an equitable way. During the pandemic, early on, most of the viral sequencing was being done in rich countries, and so there was a lack of understanding of exactly what's going on in Africa. Through work with Illumina, Africa CDC, and other partners, we were able to change that, first in South Africa, now across the continent as a whole. And so, we do see that some variants are coming out of Africa, and so both to help Africa and to have the world know what's going on, remedying this inequity is making a big difference. Of course, Africans, their genetic background is more diverse than any other population. So in terms of understanding, bad alleles, good alleles, and dealing with their particular health challenges, we need to do better of getting that information to the databases, which today are only about 1% of the information. So equity's got to guide what we're doing here. That's where our foundation has as a priority, finding exactly where those gaps are and finding the right partners to solve those things. We've got a kind of a duality here. The current health circumstances post-pandemic are not what we want them to be. It's been a gigantic setback. But on the other hand, we have this innovation, much of it underpinned by genomics and the kind of work all of you do. So on balance, I'm very excited about how we can take that capability and use it for dramatic improvements in human health all over the world. Thank you.
Thank you, Bill. What an exciting talk, and definitely a call to action. The great thing is that here in this room, we have people that have many of the tools and the responsibility to drive that genomic access for all, which is so important for us. Bill, we've heard you talk about the role of genomics and epigenomics for the analysis of infectious disease. But what do you think about non-infectious diseases? We've seen the field of oncology testing using liquid biopsy. How do you think that applications of like liquid biopsy fit in the work that the foundation is doing in global health?
Well, for rich world diseases like cancer, the rate of innovation is fantastic. There's a profit motive in terms of the drugs and the diagnosis and all that. I'm very encouraged by the progress. I tell all my friends to take the Galleria test. Why not?
Yeah, exactly.
And I do think both that test and others like it can help the detection so much that we'll actually see substantial reductions in cancer deaths, and while at the same time we're getting better at all of those treatment things. For the foundation, our basic threshold is we save lives for $1,000 per life. So when something like high blood pressure medicines get so cheap that literally by getting those out to the people who need it, you're saving an entire lifetime for $1,000, that makes sense. A lot of these cancer things aren't today in that range, but time is very helpful. I'd say a good example of this is that we have a project with the NIH to take gene therapy, which is multiple millions of dollars, clearly out of our range, and change it to be a single shot that's only a few $1,000s, because we think we can do a sickle cell cure in a lifetime, one shot, and basically our fetal hemoglobin is being expressed to avoid the problems, or an HIV cure with that shot. The reason we go for that low price is because that's how it's affordable in Africa. Now there's a huge benefit even to other health systems, at a few $1 million times a few 100,000 patients, even in the US context, that's going to hurt your ability to do other things. So, I love the broad advance in medicine driven by genomics.
Thank you. So we saw that as more sequencing comes through, we are enabling more, we lower the price. The tools that you need to do analysis become more and more complex, right? So we're starting to relationships between proteome and epigenome. How close do you think we are from applying the benefits of quantum computing to complex things like vaccine and drug design?
Well, you're certainly right that it's more complex. I mean, we hadn't paid enough attention to epigenetics. Now we're realizing that's super important for all diseases. We hadn't paid enough attention to mitochondrial DNA, and that's showing up in a number of things as being extremely important. If you look at kind of the software base that's going to help us move faster, I'd really point to the AI and machine learning, particularly as we use transformers with larger database that, as we get more and more shape data that's more and more accurate, I'm super enthused about what comes out of that. Quantum computers, I'd say that's a little less clear. People like Microsoft are putting billions of dollars into it, and at some point, they should be able to get the million or so qubits you need to model these physical systems. And so yes, I do think they may help solve some shape space things, but in the meantime, the AI transformer approach is already delivering a lot, and the pace of there is quite phenomenal.
Yeah, it has been impressive. So in your book, you talk a lot on, in today's speech, you mentioned it too. You talk about GERM, a Global Emergency Response Unit for Infectious Diseases. Can you talk about what you and the foundation are doing to make GERM a reality? And particularly, if you had to pick one thing from this audience to help you achieve your goals, what would that be?
Well, we don't want another pandemic. It's 20 million lives, $20 trillion, huge deficits, learning loss, mental stress. We're going to be digging our way out of this for a long time. And we were basically lucky. That is, the fatality rate of COVID in the general population is about 0.2%, very concentrated on the elderly. You could have another pathogen emerge, particularly if it's intentionally designed, where the fatality rate would be 40, 50%. And that's a very different thing in terms of the societal impact. So what we had was the worst thing since World War II, but it could be even worse than that. Surveillance, to me, is the obvious thing. You have to practice what you do if you spot something or how you're going to contain it. That includes scaling up the diagnostics very quickly. Countries like Australia that did that better by actually working with the people with the PCR machines had about a tenth the death rate of the United States. People thought the CDC would do that, they did not. So we're as bad as any country here. So every country needs to practice and be ready. How do you scale up diagnostics? How do you do the communications right? We do need at a global level those capacities, which is I call this global epidemic response and monitoring germ group. And the most natural place to put that is WHO. Their goal is to make sure everybody's practicing and do sometimes regional practice of how all the coordination works, because the tools will be manufactured in different places where the problem is. They also need extra capacity to help the poorest countries, because the risk of an emergence out of Africa is pretty high, and there you're not going to have the capacity. So if you have a hint of an outbreak, that global team would come in and start to gather samples, start to implement quarantine type things, with the goal that it doesn't get out to the entire world like it did this time. And it's only, it's a very modest cost to have this there. So far, the debate about, okay, what investments were we missing, both domestically and globally, starting off a little bit slow, but I'm hopeful that, the tragedy's still in our mind, and, spending a few billion to avoid having to spend trillions will begin to look like a good deal.
You think we've learned?
20 million deaths should teach us something. So
I hope so too.
We're a little slow, but yes, I think we will get a plan. One thing that slows it down, of course, the Ukraine war, the concern of the economic situation, but also for epidemiology, people look to the CDC. They're the best in the world. The fact that they didn't understand about high-scale diagnostics and ongoing surveillance, and they're just figuring out their reform, means everybody's a little bit waiting for the US to take the lead, both on the design of a good system and the resources to fund it.
Thank you. So before your pandemic preparedness book, you wrote one about climate change, and you've spoken about climate change's impact on food security. How do you think that fight is going, and is there room for genomics to help address those issues?
Yeah, agriculture is an area where I could have used that as every bit as strong examples as the human health areas that I mentioned. It's very lucky that plant genetics are the same as human genetics, because we wouldn't have invented all these tools, to sequence plant genomes if it required a separate thing. So it's kind of a trickle-down thing into the egg space. Likewise, gene editing, which is great for human health. Now we're able to improve plants in various ways far more effectively. And we can look at all the alleles and use the AI software and understand all of nature's experiments in terms of disease resistance and productivity. And as you say, this comes up in the context of climate change, because the most dramatic suffering from climate change will be poor farmers near the equator whose crops are starting to fail. Sadly, with climate change, you not only get the high temperatures, you get the water comes as floods, and then it doesn't come, you get droughts. And so the weather is way more extreme. We just saw in Pakistan, immense flooding at the same time where further east you have China in a once-in-a-century type drought. So these weather challenges mean that we have to make far, far better seeds for temperature, for basic productivity, for disease resistance, and the ability to see what's going on. That's all founded on genomics, and we're getting deep understanding into how plants resist disease and how we can put either through interference RNA or other techniques that resistance without having to change everything else. And we're getting people to understand the safety profile for that, so that African countries can make independent decisions of which of these improved seeds they should adopt.
Thank you. What do you think are the biggest dangers right now in terms of, we talked about global famine and a bunch of bad things in the world, but we're trying to learn from COVID. What do you think is the biggest danger in terms of another disease that may reach pandemic levels? Do you think it will be?
Well, there's two sources of risk. One is the naturally caused epidemic, which is basic zoonosis crossover. And the risk per year of that is much higher because the human population is so gigantic, and whether it's in meat markets with a variety of things or in Africa where we're invading these ecosystems and creating an easy chance of crossover, I'd say the risk, there's at least a 50% chance we would see another natural epidemic in the next 20 years. If we can get vaccines against the big families of respiratory diseases, either to eliminate those for the human population or to have them on standby for very rapid manufacture, then we could limit some of the damage. The other risk is bioterrorism. It's an intentionally caused thing, and sadly the tools that would let you create such a thing are improving faster than the world is setting up the kind of serious surveillance that you would need to catch something like that earlier enough. So I don't know the probabilities or anything, and that one's always a bit scary. But between the two of those, I think you can justify a little bit of preparation. I mean, we do prepare for earthquakes and fires. That's partly because there's lots of small ones, and you keep in your mind. Pandemics, we went 100 years since a gigantic one that got into the wealthiest countries. I don't think it'll be anything like that, or else we could kind of ignore it.
Hopefully, it won't be like that. You recently announced that the foundation will be increasing its annual spending from $6 billion to $9 billion a year. Can you talk a little bit about what prompted that generous increase?
Well, I'm extremely lucky that there's a significant amount of wealth that came from my work at Microsoft, and that's been well invested with good returns. And then Warren Buffett, amongst a number of foundations, he supports a very significant percentage of his wealth is going into the Gates Foundation. And so we have these resources, and when I first was thinking about, okay, I'm going to start shifting my time from full-time work at Microsoft into the foundation, I thought, well, all the previous government grants and philanthropists will have taken all the high-impact stuff, so we'll just have to come in on the margin and be a little bit creative. When I saw that there were things like malaria killing 1 million children a year, and when we gave a $30 million grant, we became the biggest funder of malaria. I was like, this is a crazy world. Why are these things not being done? And it's certainly, of all the developing countries' diseases or helping their farmers, a variety of things, the impact can be very high. And so, for me, it's fun to get to organize scientists, people who've worked in the field, policy people, partner with governments, and do this work. The fact that we have enough resources that we can up that, means we can be even more ambitious. Now, with governments, the costs they have with the Ukraine war, making sure that even as we address climate, we're not taking that money away from vaccines. The competition for the very limited aid that flows from rich countries to poor countries is more acute right now, and that total probably won't increase. So all the more reason to have innovations so that can have more impact. But it's right now, this next 5 years will be challenging. So it wasn't that I said, in 5 years we can solve all these things, but with more money and being smart about it, we can accelerate the progress.
Yeah, there is a lot to do. So it was nice to hear talking a little bit about brain research. So there continues to be major new sequencing initiatives to study the brain. And how do you think these efforts can be linked to researchers in these communities to ensure their challenges and opportunities are part of these efforts?
Well, I'd say that in the same way that genomics, the tool, is behind so much of the advance in medicine, that now various instruments that are letting us see what's going on inside the brain with better time resolution, better space resolution, or optogenetics, where we can both see and affect things. There's people who are putting stents in that can get near to cells and start to affect those. So the opaqueness of the brain is going down somewhat. So the tools are, again, kind of the leading edge of getting true understandings. The complexity of the brain, is way beyond even these other super complex things like the immune system. It is interesting that a lot of the AI techniques we're using, where we created them without studying the brain, it looks like, the brain's way of learning matches to some degree these algorithms, and so that'll be helpful for us. We don't even know the most basic thing, like how is knowledge encoded, how is it accessed. So it's early days. I put some money into Alzheimer's research, including Alzheimer's diagnostics. Some of that, the mass sequencing may help us see those signatures in the blood. And the reason that's so important is that then you can do drug trials where your endpoint is more of a biomarker than the kind of mental tests that we've had to use for the current family of antibodies. So I'm optimistic about the brain, but it is, truly the final frontier. It may take us quite a while before we fully understand it. But it's great that now researchers are inventing those tools and turning there, because the potential for improving the human condition is very, very dramatic.
Well, I thank you so much for all the work that you do and the foundation. You try to work for those that don't have a very strong voice in the world, and without people like you and the foundation, we will be in a worse place. So better is good. So thank you so much for that. Please, everybody, thank you.
Thank you.